Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting. Today we're going to continue our state review. Over the past couple of days we've been going through the biology standards and hitting all the highlights so that when we get to our state test at the end of the year, we're ready to go and we're refreshed on everything. So that's what we're doing today, and we're just going to continue moving through some of those standards. Here are the standards that we're hitting today. Today we're mostly talking about the cell cycle and how cells grow and develop as they age, and we'll talk about why that's so important for our bodies as we grow. Okay, so that's kind of the agenda today, and we'll go ahead and get the ball rolling on this. So let's go ahead and get started talking about the cell cycle. So what is the cell cycle? So I want you to imagine when you were first made, when your mother's egg and your father's sperm first came together through that process of fertilization, we make something called a zygote. So the first thing you are is a zygote, and at that point you are one cell large. But as you grow and develop, most humans today are made up of trillions of cells. So how do you start off as one cell, and then eventually as you grow older, you're made up of tons and tons, trillions of cells, right? Well, it's all through this process known as the cell cycle. As cells grow, they're going to develop, and eventually they're going to replicate. And that's how we can get one cell when you're first made to trillions of cells today. And that's all through the process of the cell cycle. And that's what we're going to be discussing here. So let's take a closer look at what's going on through the cell cycle here. So the cell cycle are the phases a cell goes through as it develops. A cell needs to perform its day-to-day activities, but also needs to replicate in order to heal wounds, to replace worn-out cells, and also to help you grow. The cell cycle is the process of getting ready to divide and to create new cells. So the cell cycle, we start off in something known as interphase, okay? Interphase, this is when a cell is not readily dividing, it's just doing its day-to-day activities. In addition to this, it will move through the following steps. This is the longest phase of the cell cycle. So the longest phase of the cell cycle is known as interphase, and we can take interphase and divide it into three smaller categories. The first part of interphase is known as the G1 phase, and that G stands for growth or gap. And what's really happening here is that the cell is just growing. At this point, the cell had just divided, so it's very small, and it's just going to spend this time getting larger and larger, okay? After that, we'll move into something known as the S phase. The S stands for synthesis, and that's really important to remember because what you need to know is that this is the point when the cell is synthesizing new DNA, which means it's making an extra copy of DNA. Also, remember that during interphase, DNA is in a form called chromatin. Okay, chromatin, I want you to imagine like a bowl of spaghetti. All those noodles just kind of tangled up with each other. That's what the chromosome, that's what the DNA looks like right now. So during interphase, the majority of the cell's life cycle, it's in a phase called chromatin. It's just that tangled spaghetti. But as we move through this cycle, eventually that's going to change. Okay, then we'll go into something called the G2 phase. Again, that G stands for growth or gap. During this time, the cell is growing, but it's a little different. Right now, during the G2 phase, the cell is getting ready to divide, and so what's really going on here is that it's doubling all its organelles. When we're done dividing, each cell is going to need its own set of organelles, so this is the time that we're making those duplicates so that when we divide, both cells have all of the organelles they need in order to operate. Okay, so here's just kind of a picture of the cell cycle. I'll just kind of walk you through it here real quickly. Okay, what's going on? This is first of all, what I want to point out is that this is a circle, right? And that's because the cell, this is a cell cycle. You're never really done with the cell cycle. As soon as you're done, you just kind of start the whole process over again. Okay, so up here you can see that this is interphase, right? Interphase is the majority of the cell's life is in interphase, and it's made up of those three smaller sections that we just talked about. So this is the G1 phase. This is when the cell is growing and getting larger. Then you go down a little bit. Right here, this is the synthesis phase, or the S phase. We're doubling all of our DNA here. This is the longest part of interphase. Remember that doubling our DNA, we want to make sure that it's right, and so that's a very long, lengthy process to make sure our DNA is correct. And then we'll go into the G2 phase, and again, that stands for growth 2. Then we'll move into something called mitosis, and that's what I'm going to spend some time talking about next. So let's move into the next part of the cell cycle known as mitosis. Okay, so let's talk about the cell cycle a little bit more. What's going on here? So after interphase, the cell's going to move into that mitosis. Okay, mitosis is division of the nucleus. Okay, this is a pretty short part of the cell cycle, but a lot of stuff is going on here, so it's important to know what's going on. Mitosis is separated into 4 different phases. Those 4 phases are called prophase, metaphase, anaphase, and telophase. And a little mnemonic to remember this is just PMAT. If you can remember the word PMAT, that'll help you remember these 4 parts of the cell cycle in the correct order. Okay? During the last phase of mitosis, so during telophase there, the cytoplasm of the cell will begin to split. And this is known as cytokinesis. And that process is a little bit different in animal versus plant cells. In animal cells, you get this thing called a cleavage furrow where the cells start to pinch off like that. That's called a cleavage furrow. And in plant cells, you get something known as a cell plate starts to develop, and it just divides the cell down the middle. So this last part, cytokinesis, is a little different in animal versus plant cells. Okay? Great, so let's take a closer look at what's going on in mitosis here. There's a couple things you need to know about each one, and I'll try to give you some key words to remember to make remembering all of those things a little bit easier, okay? So remember, we're starting over here in interphase on the far left over there. That's what the cell looks like the majority of its life. One thing I want you to notice is that the nucleus here is intact. Can you see how there's a yellow circle there in the middle and it's got the green line outside of it? So the nucleus is intact during interphase. But look what happens when we move into prophase. A couple things happen in prophase. One, the nucleus is going to dissolve. So now in prophase, we do not see that yellow circle. That the nucleus is dissolved here, okay? And that makes the chromosomes a lot easier to see. You see how the chromosomes now are they look like that X shape that we're used to seeing them in. Right now they're in the form of chromosomes, but that only happens once we get to prophase. The majority of the cell's life, it's in that form called chromatin. Okay, what else is happening during prophase? Well, look at these blue lines that are kind of spread throughout the cell. Those are called spindle fibers, and they're attaching to the chromosomes and lining them up. Kind of imagine controlling a puppet with strings. That's kind of what's going on here. We push and pull the chromosomes around using those little blue lines, which are called spindle fibers, okay? So prophase, that's where most of the stuff is happening. There's 3 things I really want you to remember. The chromosomes condense from chromatin into chromosomes. The nucleus dissolves. You see how here we have that yellow circle, now we don't. Okay, and then the spindle fibers form. So that's 3 things in prophase that I need you to remember. For metaphase, this one's a lot easier. The main thing that's happening here is that the cells are lining up, the chromosomes are lining up in the middle of the cell. Sometimes we call that the equator or the metaphase plate. But if you can just remember middle, that's going to be great here because metaphase and middle both start with the letter M. And that's going to help you remember what's going on here. During metaphase, the chromosomes get lined up in the middle. We're lining them up in the middle so that we can divide them evenly, okay? Because look what happens in the next part of mitosis is we have this phase right here called anaphase. During anaphase, you can see how those blue lines are pulling the chromosomes apart from one another, right? Half of the chromosomes are going to one side of the cell and the other half are going to the other side. Well, what I want you to remember for anaphase is the word apart, right? They're moving away or apart from one another, right? If you can remember that, you can remember what's happening in anaphase. Okay, let's go to telophase. So telophase is the last part of mitosis, and telophase is just the opposite of everything that happened in prophase. So now in telophase, we have the nucleus coming back. You see how the green dotted line is here and the yellow circles are here? So the nucleus and the nuclear envelope is coming back. Okay, the spindle fibers are retreating. So over here in prophase, you can see them growing and kind of starting to attach to the chromosomes. Now they're shrinking, right? So everything that happened in prophase, the opposite gets done in telophase. The nucleus comes back, those spindle fibers start to retreat, and our chromosomes are now uncoiling into that chromatin phase. You can see here in telophase we don't have that nice X structure anymore. They look more just kind of spread out and not as condensed, right? During telophase, around when telophase is happening, this part will happen. This is called cytokinesis, where mitosis is separation of the nucleus. Cytokinesis is separation of the cytoplasm. And so what's going on here, this is an animal cell. What's happening is the cells are starting to pinch off from one another like this and moving away. That's called a cleavage furrow in animal cells. If it's a plant cell, a wall, a cell plate just grows right down the middle and divides it, and that's called a cell plate. So the cytokinesis is a little bit different. Okay, so that's just kind of an overview of some things that are happening in mitosis. We'll just take another look at that real quick on the next slide. So this is a lot of that information that I just said. I just wanted to kind of spell it out so it's a little bit easier to read and follow along with. So let's take a closer look at this, and then we'll go ahead and start wrapping up for the day. So during mitosis, remember that there's 4 phases here. We need to know what's happening in each of these different phases. Okay, so I have prophase. Prophase is the first step. Remember PMAT. But it's also the most complicated. The most stuff is happening here. So here are 3 things I really want you to remember about prophase. 1, the nuclear envelope and nucleus is going to disappear. Most of the time that nucleus is there. It's very easy to see. During prophase it starts to dissolve. Okay, the chromosomes begin to condense. Remember that most of the time the DNA is in the form of chromatin. It looks like spaghetti. It's kind of hard to see. During prophase, the chromatin starts to coil up and turn into chromosomes. And the chromosomes are easier to see because they're thicker. That's like when you take the spaghetti and coil it around your fork before you're ready to eat it, right? And then lastly, during prophase, is that the spindle fibers begin to form. Those were those blue lines I was talking about. Okay, then let's look at metaphase. So metaphase, the chromosomes align along the equator of the cell. Just remember middle, right? The chromosomes align on the middle of the cell, middle and metaphase. That should be easy to remember. During anaphase, the chromosomes are pulled away from each other. So think away or apart, and that'll help you remember what's going on in anaphase. And like I said, telophase, this is just everything the opposite of what happened in prophase. So now the nucleus reforms, the chromosomes decondense, they turn back into that chromatin, and lastly, the spindle fibers are going to disappear. So y'all, thank you so much for making it here today. Your teacher has some other work for you to do, so make sure to give that your best effort, and I hope everybody has a great day.